We want to thank God for today. I can only see Uma Sandra. Uma Sanda. People have not keyed in. We know that our lecture is always Thursday by 3 o'clock.
Yes,
So,
sir. Good afternoon, sir.
afternoon, how are you?
Sir, I'm doing well, sir.
Okay, you are from Kano Center?
No, sir.
Where?
Sir, I'm from Benin Study Center.
Okay, Benin.
Yes, sir.
Okay, thank you. I can also see Akomaye, Margaret. Margaret Agbede. You are joining us. Thank you, Margaret. Thank you, Uma. Maybe let's revise what we did last week. From there, we now move to what we are going to do today. Last week, we started genetics. And then we started genetics already. We defined some terms we meet in genetics. Those terms that are very familiar with genetics, we look at them. Some of them include something like chromosome. We define chromosome as a rod-like substance that contains the gene. We also define the gene as a unit of inheritance. We also define heredity as a science of study of heredity and variation. We looked at variation as a tendency for the young ones to be different from their parents. Then we looked at allele, which is a pair of genes that controls a given character is called allelemorphs, while each of them is called an allele. We look at genotype as the genetic makeup of an individual. We look at phenotype as the phenotypic physical appearance of the individual. We also looked at dominant character and recessive character. Dominant characters are those characters that dominate. Recessive characters are characters that are not dominant, that are dominated. We also looked at filial generation. The offspring of an organism are what we call filial generation. We have the first filial generation, second filial generation, and third filial generation. We also looked at locus. As we said, that locus is the position occupied by gene on the chromosome. Then we also looked at homozygous condition, when the pair of genes that controls a given character are identical. That is a homozygous condition. We also looked at the heterozygous condition, when the pair of genes that controls a given character, when they are not identical. That is a heterozygous. Like capital T small t or capital R small r, this is a heterozygous condition. If you have capital T, or capital T, or capital R, or capital B, these are homozygous conditions. Now, we also looked at last week, we also looked at the shapes of chromosome or chromosome morphology, where we say we have metacentric, we have submetacentric, we have acrocentric, telocentric, and satellite chromosomes. These are what we discussed last week. Then from there, we looked at from that, we now looked at the characters variation in a population. We look at continuous variation and discontinuous variation. We say a continuous variation is when there is a gradation in the characteristics or in the character. Gradation, like we use height as for example, that if you bring 50 people, that the height is not just tall or short, no. You have those that are short, those that even shortness, you have different categories. then until you reach the tallest person. So it goes through a series. Then we also looked at height. For example, color is a continuous variation because you continue to have different gradations of color. So we also look at body weight. Body weight, different gradations of the body weight. So some people are very fat, some are not very fat, some just until you meet a very tiny person, even tiny, even those that are not fat. You have tiny, different gradations of tiny. So that is a continuous variation. Then discontinuous variation is when you have two extremes. This type of variation produces individuals showing clear-cut differences with no intermediate. In continuous variation, we have intermediate, but in discontinuous variation, we don't have intermediate. Like tongue rolling. When you roll the tongue, it is either you roll or you don't roll. You don't have any medium. You don't have any intermediate. Moving the ears. Some people can move their ear. It is either you move or you don't move. Ability to test some chemicals. It is either you test or you can't test. So these are what we call discontinuous variation. Sickle cell anemia is either you're a sickler or you are not a sickler. So this discontinuous variation. We did that last week. And we also looked at causes of variation. We looked at random fusion of gametes, gene reshuffling during meiosis, mutation. These are the causes of variation. Then we also looked at application of variation. Where can it be applied? In crime detection, in blood transfusion, in determination of paternity, in adaptation to survive. These are the various places we can apply variations. Then we looked at, last week also, we looked at transmittable characters. Characters that can be transmitted from parents to offspring. Something like what? Height. Some people, if you are so short or dwarf, you are not. It's not a miracle that you produce somebody that is tall. So height is a transmittable character. Intelligence is a transmittable character. Badness is a transmittable character. Some diseases are transmittable. So characters that can move from the parents to the offspring are transmittable characters. However, some characters are controlled by both environment and inheritance. Something like crime. You see, crime, one can get a gene that is free from crime. But then you stay in an environment where commit crime. Before you know it, you get immersed in crime. So that is it. Even intelligence, as we say, you may inherit a gene for intelligence, but you are put in a very environment that doesn't allow those genes to express, so that it will not be expressed very well. So these are characters that are product of nature and nurture. The environment controls part of the character. So these are transmittable characters. In animals, we have said it, skin color, blood grouping, fingerprints, height, facial appearance, tongue rolling, ability to taste, sickle cell anemia, hemophilia, type of earlobe, type of eye, then color blindness, diseases like AIDS that are transmittable. In plants, height also, color of flower can also be transmittable, shape of seeds, taste of fruits, size of fruits, lifespan, resistant to disease, food content of fruits, they are transmittable in plants. Even in animals, lifespan is transmittable. Some families are known to stay for a very long time. They can live up to 90, 100, 120, or 280. So while some families, hardly do they pass 70. So lifespan is transmittable. Even though the type of food you also eat can also affect the lifespan. But generally, lifespan is transmittable character. So today we are going to look at Mendelian genetics. We are going to look at Mendelian genetics. Mendel, as I told you earlier in our last lecture, did physics and mathematics in his first degree. But later on, he went and studied and joined the church in the monastery. He wants to be. He decided to join the priesthood, and so he went to monastery. And then in the monastery, he had it was, monastery is a secluded place. So, but then you have a lot of flowers to play around. So he now decided to work on some of the flowers. So he decided to cross-pollinate those flowers because he has an idea of pollination in his first degree. So he now cross-pollinated red flowered plants with white flowered plants. And they produce offspring. And surprisingly, in the first filial generation, what Mendel saw was so surprising because all the flowers in the first filial generation were all red. So he was wondering, what has happened to the white flower? Because I cross-pollinated white and what? Red. And then the first offsprings were all red. So the red became dominant character, while the white are recessive character. So he decided, it's okay. This red plant I've obtained in my first flagellation, he now crossbred them again. He crossbred that red flowered plant with another red flowered plant, that first filial generation. What he got in the second filial generation was very surprising because he no longer have all of them red. He had 75% of them red and 25% of them white. So he now got them in the ratio of 3:1. 3:1. 3/4 of them are red, while 1/4 are white. So then they began to ask, what has happened? What's really happened? So he now decided to he now did his work and kept. He now told people what, he now published what, but people do not understand. People never believed it until later in life. When people now, even when Mendel was no longer there, people now find out that that thing he discovered was very, very correct. So that was how Mendel became the father of what? Genetics, because his work stood the test of time. Now, he now propounded what we call Mendel's first law and Mendel's second law. Mendel's first law and Mendel's second law. What is Mendel's first law? Mendel's first law is called law of segregation of genes, law of separation of genes. Let us say that that red flower that is dominant now, if it is homozygous, you use capital R to represent that red. And if it is heterozygous, you use capital R, small r to represent what? The heterozygous dominant flower. So what Mendel was saying is that if we now cross this red flowered plant with a white flowered plant, and that white flowered plant is not dominant, is recessive. So capital R for the red and small r for the white. When he cross it, he produce young one, the first filial generation. All of them are capital R small r, capital R small r. All the first filial generation are heterozygous red. That means capital R small r, capital small r, capital R small r, all true. So Mendel propounded the law of segregation of genes. And what is this law? The law states that during the formation of gametes, that there is separation of genes. For example, that capital R, when it was forming gametes, one R will go to 1, the other one will go to 1. So you circle one capital R circle, another capital R circle. There is separation of genes. during the formation of gametes. That's a simple thing. The first law of Mendel's first law of inheritance, which is law of separation or segregation of genes, states that there is separation of genes during the formation of gametes. Full stop. That is Mendel's first law. So if you are using capital T, it segregates. When it segregates, one T goes to one gamete, another T goes to another gamete. If you are using capital H, when it segregates, one H goes to one gamete, another H goes to one gamete. But supposing you are using heterozygous, let's say capital T, small t. When it segregates, one capital T will go to one gamete, small, and the other second small t will go to another gamete. That is the law of separation or segregation of genes, Mendel's first law of inheritance. So if you are asked to state Mendel's first law of inheritance, your answer is now Mendel's first law of inheritance is the law of separation or segregation of genes. The law states that genes separate during the formation of gametes. You know, there is allele, two of them are called allelomorphs. Each of them is called an allele. So each of the allele enters the different gamete. Two of them can never enter one gamete. Two of the alleles can never enter one gamete. Capital T cannot enter one gamete. They must separate so that one capital T enter one gamete, another capital T enter another gamete. So the law states that there is separation of genes during the formation of gametes. That is Mendel's first law. Do we get that? Yes. We can now go to Mendel's second law. This law, Mendel's second law, is called law of independent assortment of genes. Law of independent assortment of genes. And the law states that that is segregation of a pair of factors, such as capital A, and they occur in small a, and they occur independently. So when they are separating, they are occurring independent of one another. So this one cannot control this one. Law of independent assortment of genes, that even when the genes are separating, they occur independent of one another. That the separation of these genes occur independent of one another. So it is called law of what? Independent assortment, independent separation of genes. That is the Mendel's two laws. Very, very powerful laws. The first one is the law of what? Separation of genes, that states that genes separate during the formation of gametes. The second law is law of independent assortment of genes, that says that during this separation of genes, they occur independently. That means this one does not control this one. They occur independently. So we call it law of what? Independent assortment of genes. So that is Mendel's two laws. Mendel's first law, which is law of separation, segregation of genes. The second law is law of independent assortment of genes. The first law is law of separation of genes or segregation of genes, which say that a pair of genes separates during the formation of gamete. That two allelic genes can never enter one gamete. Each of them will enter separate gamete. These two allelic genes, these two allelomorphs, they can never enter one gamete. Capital T, when they separate, one capital T go to one gamete, the other second capital T go to another gamete. So in the law of separation of genes, and the law states that two of them can never enter one gamete. If the two enters one gamete, that is a problem. That is, it will lead to what we call genetic disorder, which we are going to see later. That means there is an accident, there is a mutation, there is a genetic disorder. So that is the Mendel's first law. The Mendel's second law is the law of independent assortment, which says that there is what? That the separation of these genes occur independently. It occurs independent of one another. That is Mendel's second law of inheritance. And then we have what to call, when they now decided to do crossing of these plants, we have what to call monohybrid cross. You also have what to call dihybrid cross. In monohybrid cross, when you cross the red and the white, that is pure monohybrid, because you are only considering either red or white. But supposing when they now cross a red flowered plant with round seeds, with a white flowered plant with a wrinkled seed. You know, see, you are now considering two characters. That is what we call dihybrid. Di in Greek means two. You are now considering two characters. But mono means one. You're only considering red or white. But this time you are considering red with what? Round seed, and what? White with a wrinkled seed. So that was why it's called dihybrid cross. We even have trihybrid. For example, if you are talking of an animal, you may talk of an animal that is tall, black, and what? Heavy, big weight. The animal is tall, the animal is black, and also have what? Weight. Compared to the other animal that is short, that is fair in complexion, and thin. So you see, you are using three parallels, three characters. That is called trihybrid, and you can cross them. So you can do crossing of all these. So you have monohybrid cross, you have dihybrid cross, you have trihybrid cross, and then as time you can continue. Now we can now look at what we call back cross. Back cross. We look at what we call back cross. What is a back cross? Now, when you now cross red and white, with red being the dominant and white being the recessive, they produce the first offspring, which is what? All of them red, the first offspring. Now, supposing you pick one of the offspring and cross it back to the parent, you are doing what is called back cross. It's not very familiar in human beings because it is like taking one of your young ones and then marrying it to your father or the mother. No, we don't do it. But in animals, it can happen in animals. Like in goats, when you produce the young ones, the one of the young ones can be crossed with the parent again. This is what we call back cross. This is what we call back cross. So it is not very, in plants, it's also very, very familiar in plant. In plant, you can do back cross. In other animals, you can also do back cross. But in human being, back cross is not very, very common. Then we also do what we call test cross. I mentioned back cross, when you cross one of the offspring with back to the parent. Then what is test cross? Test, as the name suggests, you are testing the genotype of the parent. How do you test the genotype? By crossing that parent with a recessive individual. For example, the parent in the flower we are talking about can be homozygous dominant red, capital R, capital R. Or it can be homozygous, it can be heterozygous red, capital R, small r. So that's dominant. So the parent for the red flower can be capital R, small r, or it can be capital R, small r. That is the parent. So you can now test the genotype of this parent. We don't know the genotype. Assuming you don't know the genotype, because as you look at me now, you can't know my genotype. As I look at you, I cannot know the genotype. But your genotype can easily be found. Do you know how? For example, I don't know whether you are AA or AS, but this genotype can be found as easily as possible. How do you find it? By mating any of us, either AA or AS, with what? SS, with a recessive individual. If you mate with AA with SS, all the offspring will appear AS. Or all the offspring will be either carrier or normal. You won't have any secret. But when you mate that AS with SS, the result will show. 50% will be what? Normal, while 50% will be SS. It also happens in flowers. If you made RR, that pure red RR, with what? Small r, that means white. Everything there will be red. But when we make capital R with capital R, small r, that means heterozygous red. With small r, that means white, recessive white. 50% will be red, while 50% will be what? White. So when you have this 50% being normal or 50% being red, and then the other 50% being having problem, you have no alternative than to declare that the original parent is not homozygous. The original parent is heterozygous. This is what we call test cross. And so test cross means finding the genotype, the original genotype of the parent by crossing the parent with a recessive individual. When you do it, you are doing what we call test cross. But back cross is when you cross one of the offspring with any of the parents. You are not testing anything, just crossing any of the offspring with one of the parents. That is what we call back cross. But test cross is you are finding the genotype of the original parent by crossing that original parent with a recessive individual, and the result will be 50-50. Then you now declare that, look, that original parent is not a homozygous, but that original parent is a heterozygous. So that is test cross and back cross. A lot of calculations are involved in genetics, but we may not go into the calculation. A lot of calculations are there in doing genetics, but I'm not going to go into that. But what we may do, what we may well do, is that I will give you an assignment, or maybe assignment, to do this thing, to do this one I'm going to give you now. Can you do a cross between capital R, and small r? I want you to do the cross and give me the genotype of the first filial generation, the first offspring. Capital R, cross with small r. Tell me, do the crossing and tell us what the first filial generation will all be. That's the first assignment. Second assignment, cross capital R with capital R. Let us see the offspring, how it will look like. Capital R cross with capital R. That means two heterozygous crossing each other. Let's see how the result will be. You can do that. That will be a small assignment which I want you to do. So that is a cross that you can do. So remember, the parents can either be homozygous or heterozygous, or it could be recessive, as the case may be. So you must note the genotype of the parent before you can declare the answer. Now, you can calculate the phenotype, the phenotypic ratio. You can also calculate the genotypic ratio. For example, when we cross capital R with small r, all the offspring will be capital R, small r. So phenotypically, all of them are what? Red. All red. But genotypically, what are they? Genotypically, what are they? They are all capital R, small r. That's the genotype of all of them. But the phenotype of all of them, they are all red. Now, let me ask you, suppose you are crossing tall and short. For homozygous tall, capital T, cross with small t. That means the short. Remember, I didn't mix the letters, capital T, and what? Small t. I use capital T for tallness and small t for shortness. Can you do that crossing? What will be the first filial generation? What will it give us? The F1 will give us all tall. All of them will be what? Tall. That is the phenotype, phenotypic ratio. Phenotype is that they are all tall. Phenotypic ratio means all tall. Phenotype, physical appearance. But what of the genotype? What is the genetic ratio? We use capital T cross with small t, small t. What will be the genetic ratio? What will be the genotype at the first filial generation?
Capital T and small t.
Capital T, small t. It's not capital T and. It is capital T, small t. All of them. Do you understand? All of them will be capital T, small t. Can you clap for yourself for getting that one correct?
Thank
Do you
you,
understand?
sir.
Now, supposing we use height, capital H for height. Somebody that has, let's use it to represent a height, a very tall height. And small h for very small height. Do you understand? So, in the first filial generation, what will be the phenotypic ratio? All heights. All of them will be high because capital H is dominant. But what will be the genetic ratio? Capital H, small h. Now let's use black, for example, color. Black coloration, assuming that black is dominant. You use capital B for black, assuming that black is dominant. And if black is dominant, the other recessive type should be what? White. That means small b, small b. So when we cross black, which is capital B, homozygous black, capital B, with what? A recessive color, which is small b, small b. Now, the first filial generation, what will be the first filial generation? In terms of black, what will be the first filial generation?
Capital B,
small b. Capital B, small b. That is the phenotypic ratio. What of the genotypic ratio?
they are all black.
No, phenotypic ratio is that they are all black. Phenotype, physical appearance, they are all black. The genotypic ratio is capital B, small b. Do you understand? The genotypic ratio is capital B, small b. Do you understand? Okay. Now, supposing we now move to the second step, where we now use the first filial generation and cross. This time around, we are crossing capital R, small r. Can you write it down in your book? Write it down. Capital R, small r. Cross with capital R, small r. I will write it. Capital R, small r. Cross with capital R, small r. You can look at it. Capital R, small r. Cross with capital R, small r. Do that. Let me see whether you can get it. What will be the first filial generation? What will it give?
Capital R,
Do it. Let us see.
Capital R.
Yes.
Then capital R, small r.
Very good.
Capital R, small r again. Then
Very
small
good.
r, small r.
Hey, clap for yourself. Clap for yourself.
Yes.
Clap for yourself. Thank you very much. Okay. This is the result. I hope you understand. In terms of phenotype, let us look at the phenotype now. That means how they appear, whether red or white. The capital R, is it white or red?
It is red.
Red, very good. One red. The capital R, small r, is it red or white?
red.
It is red.
Another one red. Then another capital R, small r, is it red or white?
Red.
Red. One other red. Then small r, is it red or white?
White.
One white. Now count. How many of them are red? Count the number of red.
Three.
Three reds. Is to what? One white. Is it true?
Yes.
Three red is to one white. I hope you understand. Is it correct?
Yes.
That is the phenotype. I hope you understand. 3 red is to 1 white. Did you get that?
Yes, sir.
Now, how many plants are we talking about? Count the number of plants there. 1, 2, 3, 4. Is it? I hope you know it is 4.
Yes.
It's 4. 1 red, 1 red, 1 red, and 1 white. So 3 reds and 1 white. Is it true?
Yes.
So we can now say 3 red means 3 over 4 red is to 1 over 4 white. True or false?
True, sir.
Okay. You can convert this to percentage. Is it not possible to convert this to percentage? So that you can now say the percentage. So that means 3 over 4 times 100 over 1. The other one is 1 over 4 times 100 over 1. Do you understand?
Sorry, sir. Please, what's the topic?
3 over, I'll say, want us to convert this into percentage. We have got 3 red is to 1 white. That means 3 over 4 red is to 1 over 4 white. I say convert that 3 over 4 red to percentage. That means 3 over 4 times 100 over 1. Can you calculate? 75%. Very good. So we
And
now have 75% red. Are you following me?
Yes.
Is to
Good evening, sir.
the other one. Calculate the other one. 1 over 4 times 100.
25%.
25% what? White.
White.
Do you understand?
Yes.
So the
Good
phenotypic
evening, sir.
ratio
Please, I'm
now,
just joining.
the
What's the topic?
phenotypic ratio now is 3 is to 1, or 3 red is to 1 white, or 75% red to 25% white. Do you get that? Did you
Good
get
evening,
that?
sir. Please, I'm just joining.
Yes,
What's
sir.
the topic?
Okay. Thank you very much. 3 over 4 red is to 1 over 4 white, or 75% red is to 25% white. That is the phenotypic ratio. Now, let us look at the genotypic ratio, the gene now, the genotypic ratio, the genotype ratio, the genotype. Now, capital R, we are going back. Capital R, capital R. How many of it in that place? Capital R, capital 1. So 1 capital R, capital R. Then is to, you put semicolon, is to, how many of them are capital R, small r?
2.
So 2 capital R, small r. Then another semicolon is to, how many of them are small r, small r?
1.
1. 1 small r, small r. Are you following me? Are you following me?
Yes, sir.
Now, you can now say for capital R, 1, I think it is 1. For capital R, small r, they are 2. And for small r, they are 1. Is it true?
Yes, sir.
So you can now say 1 over 4 capital R, 2 over 4 capital R, small r, and 1 over 4 small r, small r. Do you get that?
Yes.
Yes.
Just as we did for the phenotype, we are doing for the genotype. Do you understand? So the genotype ratio is 1 is to 2 is to 1, or 1 over 4 is to 2 over 4 is to 1 over 4. Do you know you can convert this to percentage?
Yes, sir.
Okay. The first one, the capital R is 1 over 4 times 100 over 1. What is the percentage? 25%. Capital R is to 2 over 4 times 100 over 1.
50%.
50% capital R, small r is to 1 over 4 times 100. 1 over 4 times 100, what is it?
25%.
Yes. So the genetic ratio is 25% capital R, 50% capital R, small r, and 25% small r, or 1 is to 2 is to 1. Clap for yourself. I didn't hear your clap. Clap for yourself. Okay. So it's very, very important. That is how it is. So so we use R now. Suppose we now use tallness. If we use tallness, can we use tallness now to do example? Where I asked you, a homozygous tall man was crossed with a homozygous short man, where the tallness is represented by capital T, while the shortness is represented by small t, small t. Can you do the crossing to find the first filial generation? Can we do the crossing? A homozygous tall man, capital T, cross with a homozygous short man, small t, small t. Capital T, cross with small t, small t. Can you do that? Now, do the crossing.
So they will all be heterozygous tall.
That's capital T, small t, capital T, small t, capital T, small t, and capital T, small t. They are all heterozygous what? Tall. That is the phenotype. Phenotype. All tall. Is it true? Is it true? Now,
Yes,
what of the genotype? What of the genotype? The genotype ratio. All what? Capital T, small t.
small t.
Yes, all capital T, small t. Very good. So that is it. Now, supposing you now have heterozygous tall man crossed with a heterozygous tall man crossed with a heterozygous tall man. Capital T, small t crossed with capital T, small t. Can you do that? Can you do that? Capital T, small t cross with capital T, small t. Just as we do it, capital R, small r, cross with capital R, small r. Same thing. So do the crossing. Let us see the phenotype and the genotype. Can you do the crossing?
Yes, sir. We're on it.
Start doing it.
Capital C, capital C.
Yes.
Capital C, small c.
Yes.
Small c, small c. Capital C, small c, then
small t, small t. Very good. Clap for yourself. Now, the phenotype. Okay, thank you. What will be the phenotype ratio now? Physical appearance, phenotype ratio. How many tall?
We have 3 tall.
3 tall. Short. We have 3 tall and what?
1 short.
1 short. That means 3/4 tall and 1/4 short. I hope you understand. Which is the same thing as 75% tall and what?
25% short.
25% short. Thank you very much. Then let's look at the genotypic ratio, the genotype ratio. The genotype ratio. Capital T, how many of them?
I have 2 of them.
Capital T, capital T.
Just
how many? 1. 1 capital T, comma, or is to, semicolon, is to. Capital T, small t, how many of them?
2.
2 capital T, small t. Very good. Then small t, how many of them?
That's 1.
1, small t, small t. Then the genotypic ratio is 1:2:1. 1 capital T, 2 capital T, small t, and 1 small t, small t. In other words, 1/4 capital T, 2/4 capital T, small t, and 1/4 small t, small t. Which also means 1/4 capital T means 25%. 25% capital T. Then 2/4 capital T, small t means what? 50% capital T, small t. And then 1/4 small t means what? 25% small t, small t. Did you get that? Did you get that?
Yes, sir.
That's very, very fine. So we have been able to do something today. We have been able to do monohybrid cross and then dihybrid cross. The first one we did is monohybrid. Okay, all the things we did today is monohybrid. When we come next time, we are going to look at dihybrid. So let us do a kind of summary of what we did today. Today we looked at Mendelian genetics, Mendel's first law of inheritance, which is the law of separation or segregation of genes. And the law states that two allelic genes will never enter one gamete. The law states that genes separate during the formation of gametes. That's it. That's the law of separation or segregation of genes that states that there is separation of genes during the formation of gametes. And two allelic genes will never enter one gamete. They will enter each gamete. They can never enter one. They must enter separate gametes. That is the Mendel's first law. And the Mendel's second law is law of independent assortment of genes, which says that during it says that there is segregation or separation of that the separation of genes occur independent of one another. During this separation, they occur independent of one another. So that is what we learned today. First law of Mendel's first law and Mendel's second law. And then we saw monohybrid cross. When you are dealing with one character like red and what? White, tall and what? Short, big stroke small. This black stroke what? White. This is a monohybrid cross, which you have done today. Then you have dihybrid cross. When you combine two different traits, for example, somebody that is a flower that is red with round fruit, or round seeds, with another plant that is white and wrinkled seeds. So you are giving two different characters together. You are taking them together. That is dihybrid cross. Then you also have trihybrid. You are taking three characters at the same time. Maybe red flowered plant with wrinkled seeds and then round fruits. Three things. So, or a man that is tall, black, and what? Huge. You are taking three things at the same time. So that one is trihybrid cross. Then we now enter to do some calculations, some simple calculation in the monohybrid cross. Like when you use capital R, small r, and capital R, and small r. In the first filial generation, all of them will be red. Then the second filial generation, we cross capital R and small r. Then we have the phenotypic ratio as 3/4 red as to 1/4 white. Then we now do the genotypic ratio of this second filial generation, which is 1:2:1, or 1/4 red is to 2/4 red, heterozygous red, 1/4 capital R, 2/4 capital R small r, and 1/4 small r. These are some of the things we did using R. We also use T to represent. We did the example with T. For example, a tall man marrying a short man, capital T, going with small t. In the phenotype of the first filial generation, everything is tall. Then the genotype of this first filial generation, everything is capital T, small t. Then they now cross again, capital T, small t, cross with capital T, small t. The phenotypic ratio reads 3:1, or 3 tall is to 1 short, or 3/4 tall is to 1/4 short, or 75% tall and 25% short. Then we also looked at the genotypic ratio, which is 1 capital T, 2 capital T, small t, and 1 small t, which is 1/4 capital T, 2/4 capital T, small t, or 1/4 small t, or 25% tall is to 50% tall, or 25% capital T, 50% capital T, small t, and 25% multi. These are the things we studied today. And then in our subsequent lecture, we are going to look at some other things concerning maybe population genetics or thereabout. So do you have questions? If you have questions, you ask me. Kehinde, you just came in. So ask your questions. If you have any questions, I can see Michael Etim. I can see Matthew Agaba. I can see Benjamin, Peppo, Dennis, Badmon, Solomon, Bamidele, and more others. Ayu, Christopher, Rose. If you have any question, ask. Let's take it from there. You don't have question?
I've been here before. I just went to do something.
Okay. Thank you very much. Thank you. Bamidele, do you have any question? Or Benjamin? No question? Okay, which means you all got what we did today. Matthew, you want to ask question? Yes.
Yes, I want to ask a question. Good afternoon, sir.
Afternoon, Matthew. How are you?
Fine, sir. So we have really missed your facilitation. So our hands have been on practical all those while. We just finished on the 30th. And we've missed all your class. The question I want to ask is this topic you treated today, which unit and what topic is this, sir?
Genetics. We are talking of Mendelian genetics. Very, very important. Very important.
Unit? It's under which unit?
You say under which unit? Under genetics.
Okay, under genetics. Okay.
Yes.
All right, thank you.
So what we may do is maybe next week we use half of the hour to repeat this so that all of you can follow.
Okay, sir. Okay, because a lot of us are not aware. We've been on practical, that's why.
But our lecture is every Thursday by 3 o'clock.
Yes, sir. The practical, it was tough for us. So we'll go in the morning and come back at night.
It's okay. Thank you very much. Thank you. Any other question? Any person that has question? Okay. Without any other question, I say thank you very much and hope we meet next week to summarize all these things so that all of you can follow. Is that okay? I think we are coming towards the end of our lecture. Please, starting from the first lecture, go through. I will post this back. I will post it in our this thing. I will paste it so that you can go through it and rehearse it and look through my lectures so that you grasp it. I'm going to paste it for you. So thank you and God bless you. This is where we'll call it. We'll come to the end of the lecture. Thanks and God bless you.